Okay, so next we are going to move on to module 36, and this module is looking at prenatal development. So this is from the time that conception occurs until birth occurs. So this is all pre-birth at this point, okay? And there are two questions that we're going to answer. The first one is, what is the nature of development before birth? The nature of development before birth. And then the second question is, what factors can affect a child during the mother's pregnancy? So is there anything the mother could do while she is pregnant that can actually affect the child? That's what that question is going to look at. All right, so let's look at something really basic, and we're going to talk about genetics first off. So this is going back to probably like a biology 101 course. This is stuff that you probably learned in your high school biology class or high school health class. All right, so first we're going to talk about chromosomes. A chromosome is a rod-shaped structure that contains all of your hereditary information. Contains all of your hereditary information. Along with chromosomes, we have your genes or your genetic makeup. This is part of the chromosome through which the genetic information is transmitted. So you have the chromosomes, and whatever genetic information or hereditary information is in those chromosomes, it is transmitted by way of genes. These are composed by a sequence of DNA. DNA controls the development of systems of the human species. So it controls the development of the heart, the circulatory system, the brains, the lungs, and so forth. We'll talk about this physiological development in just a bit. But it is your DNA genetic makeup that controls the development of these various systems. It also shapes the characteristics that makes each of us unique and different from each other. The only individuals that have the exact same DNA, the exact same genetic makeup, are identical twins. Everyone else is completely different in their DNA than another individual is. It also determines the child's sex by a particular combination. And these combinations are 2 X chromosomes for females, and both an X and a Y chromosome develops as a male. So you get the X chromosome from your mother. You get either an X or Y chromosome from your father. So your father actually determines the sex of the fetus. So XX is a female, XY is a male. So this picture, figure 1, this is at the top of page 378. And here you can see conception, right? That is the sperm entering, and it will connect with an egg. So this is the moment of conception. In letter B's figure, it is humans receive 23 pairs of chromosomes, half of them from the mother and half of them from the father. Okay, 23 pairs of chromosomes, half from your mother, half from your father. Letter C figure, that's your DNA sequence. And in letter D is each chromosome contains thousands of genes that actually help in construction of future development of the body. So this is going through and showing you exactly what happens. So conception, you then get 23 pairs of chromosomes, one pair from your mother, one pair from your father. These then help in this sequence. The chromosomes make up the sequence of DNA, and then each chromosome contains thousands of genes, and that helps with your genetic makeup. This figure is at the bottom of page 378. And this is what I said previously whenever I was talking about the male and female and determining. So you can see that the mother gives an X chromosome to a baby girl and an X chromosome to a baby boy. The father either gives an X or a Y. So if the father gives an X chromosome to the baby girl, that's two X's, and that means a female. If he gives a Y, then that's an XY, and that is a male. So the mother always will give X's, and the father will give either an X or a Y in part of those 23 pairs of chromosomes. And so it is the father, again, that determines the sex of the child. At the beginning of this century, in 2000-2001, scientists started what was known as the Human Genome Project. And this Human Genome Project was able to map the specific location and sequence of every human gene in 2001. That's the Human Genome Project. They were able to map the specific location and sequence of every human gene. And this Human Genome Project actually started this revolution in healthcare. Because now, because of the Human Genome Project, scientists can identify the particular genes that are responsible for genetically caused disorders. So this was like, mind explosion. That because we were able to identify and see the location of every human gene, we now can use that information for looking at various genetically caused disorders. You can also look at the various risk factors in children that can be identified because of locating and identifying all these genes. And this actually led now to new treatments for both physical and psychological disorders that have now been made possible. Before the Human Genome Project, we were not able to do that. So this Human Genome Project in 2001 has just opened up so many doors, not actually in the prevention of disorders, but more so the treatment of disorders. Okay, so now what I want to do is I want to actually look at prenatal development. And I want to start with the earliest development that occurs, starting with conception. So you have a zygote. This is a new cell formed by the union of an egg and a sperm. So at conception, when the egg and the sperm combine, it develops a zygote, which is this new cell. This occurs in the first 2 weeks after conception, and it is known as the germinal period. The first 2 weeks following conception is known as the germinal period. From week 2 through week 8, it's known as the embryonic period. So week 2 through 2 months, it is known as the embryonic period. And so now that zygote turns into an embryo. That embryo has a heart, a brain, and other organs. So it starts whenever the sperm and egg get together, a zygote is formed. That's the first 2 weeks, the germinal period. Then week 2 through week 8 is the embryonic period, and that's where that zygote now turns into an embryo. And you have a heart, a brain, and other organs. These organs are clearly recognizable despite being at a primitive stage of development. Now, from week 8 until birth, this is known as the fetal period. So you have the germinal period, the embryonic period, and now the fetal period. 2 months until birth is the fetal period. So now that embryo becomes a fetus. So at 8 weeks after conception until birth, you now have a fetus. And this is when the movements of that fetus become strong enough that the mother can actually detect them. She can sense those movements of the fetus. You actually have this age of viability. And this is the point at which a fetus can survive if born prematurely. And this occurs prenatally, age 22 weeks. So about 22 weeks after conception, if that fetus was born, it could survive. It's the age then of viability. Now, when we talk about prenatal development involves passing through several sensitive periods. What we mean by sensitive periods is that it is a time when organisms are susceptible to certain kinds of stimuli. They are susceptible to certain kinds of stimuli. And these sensitive periods can also occur after the fetus is born. So let's talk about some of these sensitive periods. You can see here on the left at the first arrow, at the end of the normal 38 weeks of pregnancy, the fetus typically weighs 7 pounds and is about 20 inches long. Then preterm infants are born before 38 weeks. Because they were unable to develop in utero, they are at a higher risk for illness, future problems, and possibly also death. So the best case scenario is to go the entire 38 weeks. However, if you are born prior to that 38 weeks, you are a preterm baby. And that leads to you could have many different difficulties with that. Now, let's come back, and we're still talking about the fetus. But let's talk about the genetic influences on the fetus. In 2% to 5% of births, the child may have serious defects. And this is a major cause. The major cause is because of faulty genes or chromosomes. 2 to 5% of births, the children have serious defects. And one of the major causes is faulty genes or chromosomes. One type of defect that you could have is called PKU. PKU is where you cannot produce an enzyme required for normal development. Fetus could also have sickle cell anemia. Sickle cell anemia. This is abnormally shaped red blood cells that may be passed on by about 10% of the African American population. So you tend to see sickle cell anemia more often in African American babies than what you would in Caucasian or other babies. Another type of heredity disability is Tay-Sachs disease. With individuals who are born with Tay-Sachs disease, they usually die by age 3 or 4. And this is because their body cannot break down fat. And this is most often seen in the Jewish population of Eastern European ancestry. The fourth type of genetic disability is Down syndrome. This is a cause of intellectual disability in which the zygote receives an extra chromosome. So it increases the risk with mothers, or this is an increased risk of Down syndrome with mothers who are over the age of 35. If you are female and you get pregnant after the age of 35, there is an increased risk of you having a child with Down syndrome. So these are four types of genetic disabilities that the fetus could have: PKU, sickle cell anemia, Tay-Sachs disease, and Down syndrome. Now, the next slide, this is going to look at prenatal environmental influences. And the one thing we're going to look at is teratogens. Teratogens are environmental agents such as drugs or chemicals, a virus, or another factor that produces some sort of birth defect. So if the mother, while she is pregnant, does drugs, that could be a teratogen. If she consumes alcohol, that could be a teratogen. If she has a virus while she is pregnant, that is a teratogen. Or there could be another factor, but all of these teratogens can produce various birth defects. Among the major prenatal environment influences are the mother's nutrition, the mother's illness, the mother's emotional state, and the mother's use of drugs, alcohol, and or nicotine. These are all types of teratogens that can influence the fetus while it is still in the womb. Even mothers who consume very small amounts of alcohol during pregnancy, they place their child at risk for fetal alcohol syndrome disorder, or FASD. Fetal alcohol syndrome disorder is now the primary preventable cause of intellectual disability. Just don't drink while you're pregnant, and you have a lot less risk at having a child with an intellectual disability. This is figure 3 in your textbook, and this you can find at the top of page 382 in your textbook. And it's going to show various environmental factors and in the possible effects on prenatal development. So rubella or German measles, if the mother contracts that, it can cause blindness and deafness, heart abnormalities, and even a stillbirth. If the mother has syphilis, intellectual disabilities, physical deformities, and maternal miscarriage. If the mother is on addictive drugs, that can lead to a low birth weight. The infant could be born addicted to the drug also. And that then could lead to a possible death after the infant is born because of going through withdrawal symptoms. If a mother smokes cigarettes or any sort of nicotine, that can lead to premature birth or low birth weight and length. If mother consumes alcohol, intellectual disabilities, lower than average birth weight, a small head, and limb deformities. If the mother receives radiation from x-rays, that's why anytime you have an x-ray, they always ask a female if there's any chance she could be pregnant, because that could lead to physical deformities and intellectual abilities. An inadequate diet, the mother's age, younger than 18 at the birth of a child. This can actually lead to a premature birth or increased incidence of Down syndrome. The mother's age, if it is over the age of 5, that could lead to an increased risk of Down syndrome. AIDS and Accutane, various other things that could lead to various physical or intellectual disabilities in the child after they are born. The last slide in this module is looking to alternative paths of conception. So let's say that a male and female are having trouble conceiving a child. And so there are alternative paths that they could seek out in order to have children. So scientists have devised several remedies to overcome infertility. And we have 3 different types that we're going to discuss on this slide. The first one is in vitro fertilization, or IVF. This is where the male's sperm is used to fertilize the woman's egg, but that's actually done in a laboratory. The fertilized egg is implanted then into the woman's uterus. So you take sperms from a male and you take eggs from a female and you have them conceive, you have them come together, but in a petri dish. And then that egg is inserted, the fertilized egg is inserted into the woman's uterus. You can also have a second type of alternative path, and this is GIFT. This is gamete intrafallopian transfer, and also zygote intrafallopian transfer, or ZIFT. So you can have the GIFT or the ZIFT, whether it's a gamete or a zygote. And what happens here is an egg and a sperm or fertilized egg are implanted in a woman's fallopian tubes. So the in vitro fertilization is where the fertilized egg was implanted into the woman's uterus. With the GIFT or the ZIFT, the fertilized egg is implanted into the woman's fallopian tubes. Now, the third type path to conception is also surrogacy. And this is where the fertilized eggs are implanted into another woman who agrees to carry the child to term. So it would not be the mother that the fertilized egg is implanted to, but it is another woman. And she's actually going to carry that child to term. So these are 3 different paths to conception. Now you may be saying, well, what about adoption? Yes, adoption is another path to having a child, but it is not a path to conception. That child was already born and now you are adopting them. Conception is starting with the sperm and the egg leading to a fertilized egg being implanted into a woman.